Hello everyone. Today, we are going to introduce an article titled The Pivotal Role of Rad51, the literature review on the bridge connecting DNA repair, tumorigenesis, and immune regulation was jointly completed by Qin Zhendong, Lu Minjun, Guo Jingwei, Wang Chenyan, Ma Ting and others from the Cancer Research Center of Beijing Chest Hospital, Capital University, and the Beijing Institute of Tuberculosis and Thoracic Tumor. This review has been published in the journal Med Research. This picture summarizes the main DNA repair pathways of cells and demonstrates the sophisticated defense network constructed by the body to maintain genomic stability. When DNA suffers different types of damage, cells activate highly specific repair tools in the face of single base damage or single-strand breaks. Base excision repair and nucleotide excision repair are responsible for handling the situation. For the most fatal DNA double-strand breaks, cells have prepared multiple pathways: high-fidelity homologous recombination repair, rapid but error-prone non-homologous end ligation, and single-strand annealing to handle complex breaks. This entire system works in coordination, forming a strong defense line that maintains the stability of our genome. The Rad51-mediated homologous recombination repair pathway is an important mechanism for cells to respond to DNA double-strand breaks. These fractures may result from endogenous stress or exogenous injury. During this repair process, the mRNA complex first recognizes DNA double-strand breaks and activates the key kinase AT. Then, the single strand of DNA at the fracture site will be wrapped and protected by RPA proteins. Then, under the influence of multiple regulatory factors, Rad51 begins to assemble into riboprotein filaments. Among them, it is regulated by positive-negative regulating factors. Finally, an intermediate structure known as steering will be formed and repaired through three possible pathways. In normal BRCA-positive cells, the PARP protein is responsible for repairing single-strand breaks, while the complete homologous recombination pathway can effectively repair double-strand breaks, thus enabling the cells to survive. However, in BRCA-negative tumor cells, PARP inhibitors not only block the repair of single-strand breaks but also firmly lock PARP protein onto the DNA, which eventually leads to the damage evolving into fatal double-strand breaks. A sophisticated pathway that connects DNA damage with immune alerts, the CGAS sting signaling pathway. When DNA, including that related to Rad51, appears in the cytoplasm, CGAS is activated in a manganese-dependent manner. The activated CGAS then synthesizes the second messenger cGMP, which in turn activates the STING protein. Immediately after, STING triggers the phosphorylation of TBKE kinase, promoting the entry of transcription factors IRAF-C and NF-kappa B into the cell nucleus, thereby initiating the production of key immune molecules such as type I interferons and interleukins. Rad51 plays a core role in homologous recombination repair. Its double-edged sword effect is reflected in maintaining genomic stability while driving tumor progression by promoting tumor drug resistance related to EMT, replication fork stability and multiple pathways, and regulating the immune microenvironment affecting CGAS sting, neoantigens, and PL1. Small molecule inhibitors targeting Rad51, combined with immunotherapy, liquid biopsy, and artificial intelligence prediction models, have become a highly promising precision tumor treatment strategy. Although significant progress has been made in the role of Rad51 in tumor DNA repair and immune regulation, key scientific issues such as its specific functions in different tumor types, the spatiotemporal dynamics of expression regulation, and the interaction mechanism with tumor microenvironment cells still need to be further explored. Future research shows on the selective delivery of Rad51 inhibitors, the precise screening of biomarkers, and the synergistic mechanism with other targeted drugs to promote their clinical translational application.